My name is Mark Denham. I'm a group leader at the Department of Molecular Medicine at Aarhus University Hospital. And today I wanted to share our project Appetite, which is together with Novo Nordisk. And for this project, we're reconstructing the human reward circuit to tackle obesity-induced cardiovascular disease and cardiometabolic disease. So obesity affects over 1 billion people around the world and costs over $180 billion annually. And we know that it's the upstream driver of cardiovascular and cardiometabolic disease. And obesity is a complex disorder where all of the treatments are focused on the symptoms. And that includes these new GLP-1 receptor agonists that can slow gastric emptying and treat obesity. However, we know that these GLP-1 receptor agonists can also act on some of the neurons involved in the reward circuit, such as these NTS and AP neurons. And it's the effect on some of these neural populations that can induce this nausea side effects from these GLP-1 receptor agonists. But that also means that we could potentially develop new drugs that could affect specific populations of neurons that then would bypass this nausea effects and still have this benefits of similar to that of these GLP-1 receptor agonists. And so in order to investigate these new potential drugs or to develop platforms for developing these neural drugs, our lab has generated this pluripotent stem cell platform that generates mycos, which are these miniaturized control organoids. Where using this platform, we can generate different brain regions, such as the forebrain, midbrain, or hindbrain region. And in particular, this hindbrain region is where these neural populations involved in this reward circuit derive from, this NTS and AP neurons. And so by generating different populations, such as these VTA dopaminergic populations within the midbrain, which is also essential in this reward pathway, we can then generate these co-culture systems and culture them together to then create this in vitro reward circuit. And using this reward circuit, we can then start to investigate new drugs that can then affect different subpopulations. And so for this project, the contribution from my lab are this pluripotent stem cell differentiation systems where we can generate these co-culture reward circuits. And then Novo Nordisk is using their CRISPR engineering technology to engineer in different reporters into these neural populations where we can then focus on specific neural reward circuits to then start to have some nice readouts using calcium imaging for looking at the activity of these neural populations. And so what we're looking for are collaborators from both the industry and also the research groups. So on the industry side, this could be any type of industry that's interested in this platform where they could be a part of helping to develop the platform, where they can use this outside of ODIN to screen for their own drugs that may be important for different aspects of this reward pathway. Could also be other industry partners involved in neuroinflammation or neurodegeneration, where they're interested in looking at human neural populations. Or it could also be research groups that have patient-specific samples that carry known obesity-linked gene variants. Or we're also very interested in groups that have good calcium imaging capabilities that could complement these two aspects that Novo Nordisk and my group brings. And also we're interested in groups that might be have strong expertise in blood-brain barrier that we could also incorporate into this model system. So if you're interested, you can of course contact me on the details below and would be happy to talk further. Thank you.